Oops, sorry, I have problems changing the slides. Okay, so I attended a webinar by Genomics Alberta last Friday, and it was mentioned that many people still don't know what genomics is, what it's really about, what it really means. So, and they confuse it with gene manipulation, which it isn't. So genomics is the science of understanding, interpreting, and harnessing DNA codes to create real-world solutions. So it's about obtaining and analyzing valuable data and using it to create innovative solutions applicable to concrete problems and, or situations in different fields. So such as human health, agriculture, forestry, environment, mining, energy, etc. So this is the power of genomics. It can be applied very largely. So one of the most powerful genomics tools is high throughput DNA sequencing, which can generate large amounts of data and very efficiently. And we can sequence whole genomes, or we can characterize a microbial population by using a common gene. So it allowed scientists to move from low-tech microbiology techniques to high-tech genomics tools, and it has given them access to a whole new perspective on life systems. So next generation sequencing techniques, they are evolving very fast. So here on this slide, we can see that the cost of sequencing technologies has drastically dropped over the last decade. So medical research and the goal to fully sequence the human genome, it drove companies to innovate and engineer new sequencing platforms with new chemistries to produce more data, and also with a lower error rate and better quality and longer reads for easier genome assembly. So on the slide from NIH, we can see the cost of sequencing a human genome has dropped from close to $10 million to about $1,000 over a decade. So this is why these tools can now be applied to environmental research. This is why they are more widely used and why we have heard more about them within the last few years. Before, it was unthinkable to spend that much money to analyze environmental samples. Obviously, pharmaceutical and environmental research, they don't share the same budgets. So, but to fully exploit the potential of these next generation sequencing tools, there's also a need to develop some better compute resources to handle and to analyze the data sets, which keep getting larger and larger and more complex. So when you calculate the final cost of sequencing a genome or a sample, you should also take into account the compute resources that are needed and the analysis by a bioinformatician. So here, I would like to discuss one of the newest sequencing platforms by Oxford Nanopore. It's called the MinION, and it's a portable sequencing device. It's a little larger than a USB, a little bit smaller than a stapler, and it can actually be plugged into a laptop to generate some sequencing data. So it's using streamlined protocols for sample preparation and sequencing, which means all of the steps can be performed outside the lab facility, and the results can be obtained fast. So this could be a good application for mining industry because there's no need to ship the samples. You can perform all the steps on the field, because as we know, mining sites are mostly remote sites, and shipping samples to a lab facility, it takes time. The samples can be degraded if they're not preserved properly. So there's lots of time between sampling and obtaining a result that can be actually biased. Whereas if you do everything on the field, you get a faster, maybe more accurate result. So this company is very active and launching products frequently to improve the results and to offer new possibilities. For instance, there is a new flow cell for sequencing that will be generating less data, but it will also be 10 times cheaper. So it will be great, for instance, for analyzing single samples. So you can perform more frequent analysis and do some monitoring. like testing water samples or monitoring a bioreactor that's performing biomining, bioleaching. And there's also another product that will be out pretty soon, which is called the SMIDGion. It's even smaller than the MinION, and you'll be able to plug it on your cell phone to perform DNA sequencing. So although it all sounds like science fiction right now, it's probably only a few years away before these new applications are widely used in different fields. It's very exciting. So now let's talk about the mining microbiome. So a microbiome is a community of microorganisms inhabiting a particular environment. So I'm sure you've all heard about the human microbiome, which has been discussed a lot over the past few years. And now in the scientific literature, the gut microbiome is considered an organ, since the human body just wouldn't be able to function normally without it. So with all that knowledge that we have acquired on the microbial populations in the gut, their interactions with probiotics, curing infections and medical conditions through gut microbiome replacement, we can all transpose this to other situations in environmental research field. So we could use some valuable information to develop more efficient and targeted innovative mining practices using the genomics tools. So why can we talk about the mining microbiome? Well, microbes, they can be found everywhere on a mining site. They are present on rocks where they interact with the minerals and can even contribute to their formation. The mineral microbiome present on the rocks is a rich source of genetic information on how these microbes could be used to extract, to concentrate, to transform the metals. So we should take advantage of this information and use it. Also, on a mining site, in the environment, in the water, in the soil, there are microbes that contribute to the soil health and plant growth. And in tailings, some microbes can actually grow and in these very harsh conditions, and they can contribute to the acid mine drainage. So the applications of genomics. So everywhere you can find microbes and biology, you can actually use genomics tools. The genomics tools, they can be applied throughout the mine life cycle. So the data can be used to complete some geochemical characterization during the exploration phase. And also during exploration, some of the bacteria, they can sequester metals and they can be used as highly sensitive detectors. And some of the genomics tools could be used to generate maps of key microbial groups. So integrating microbial ecologists to exploration teams would make a lot of sense and could open up new possibilities. This is missing from the industry, actually. In conferences and workshops, whenever they discuss the different experts required for operating mines or designing some tailings facilities, we never see microbiologists or microbial ecologists. However, there are microbes all over mining sites, and we need to characterize them and monitor their activity to optimize practices. So genomics tools can be used to monitor soil microbes, which are indicators of soil health, and we can work towards prevention rather than damage control. So there are many factors that affect the microbial community diversity and composition, such as metal contamination, changes in pH, removal of the vegetation cover. So soil microbiome monitoring is a great prevention tool. We can also, and it's also very linked to the remediation at closure, because the microbes, they play a role in bioremediation and also in phytoremediation through their interactions with roots. They can help to promote plant growth on the poor soils, which soils are usually affected after the mining activities, have less nutrients, and microbes can help plants to grow in these conditions. We can also use genomics tools to monitor and to improve some bioprocesses like biomining and bioleaching, and we can use them to treat and prevent acid mine drainage. So concretely, what kind of information do we get from the genomics analysis? So we can monitor and characterize the microbial populations in a sample. We can obtain some information on taxonomy, so who they are, who are the microbes, and gene function, what they do, what are their metabolic functions. We can also further study some relevant metabolic activities by monitoring specific genes under different conditions, and we can even quantify the abundance of the genes. So we get a gene copy number per nanogram of DNA, and we can follow the increase or decrease of the gene copy number under different conditions. So for instance, if we run a time course study, we could see the variations in gene copy numbers over time, or we can sample, we can have different sampling points on a mining site, and then we can observe the gene copy number variations at the different sampling points, and also using other metadata such as soil pH, metal concentrations, we could correlate the gene copy numbers to the metadata. Genomics tools, also the DNA sequencing, it gives us access to some genetic information on uncultured microbes, which was not possible before with the traditional microbiology methods. So we're able to access the genomes of the uncultured bugs. And this, the DNA sequence, it gives us some information on the growth conditions that we could try to grow them in the lab. Sometimes it's still necessary to grow them because we need to study further the microbes, or we need to use them for different bioprocesses. So in this case, there's a new tool that has been developed, the iChip technology, and it's to mimic the environmental conditions that the microbes usually grow under. And then after a few rounds of these of growth in the iChip, we can actually domesticate the microbes and grow them under lab conditions. So these are all genomics tools that can really make a big difference in the different projects. So here I have an example of a stacked bar graph that we can plot using DNA sequencing data. So here each color represents a different bacterial genera. On this particular graph, we are comparing samples from natural sites versus disturbed sites. So we can see which genera are the most abundant under the different conditions. And then we can also plot the same data set in different graphs to better visualize the effect of a particular parameter that we're interested in. So on the left, we have a fancy Venn diagram, and on the right, we have a principal coordinates analysis plot. So this kind of plot, it shows us, like in this plot, the samples that show the most similarity will cluster together, which means it's easier to observe which conditions affect the microbial population composition or the gene function. So there are a lot of different visualization tools that can be used to interpret the DNA sequencing data. So in conclusion, genomics is a powerful tool that has been underutilized in the mining industry. It can be used anywhere where there is biology in a mine, and there is a lot, believe me. It provides innovative alternatives for metal exploration, extraction, concentration, and transformation. It could allow extraction of low-grade ores or tailings reprocessing, which means value from waste, creating value from waste. Applications could complement or replace existing analysis or processes with economic and environmental benefits. And it could reduce mining's footprint by providing good tools for site characterization, monitoring and remediation, as well as waste management and valorization. So we could also get a better understanding and better characterization of mining sites using the genomics tools, for instance, by studying baseline diversity of microbes And this eventually could accelerate permitting and improve social acceptability of projects. But we need to bring the regulators at the discussion table to make sure that they understand and approve these tools. And we also need to include the different communities living around the mining sites. And I would also like to mention that Genome Canada will be launching a large-scale applied research project in January 2020. It's a funding opportunity that will support projects focused on applying genomics to Canada's natural resource sector. And they want to build teams with people from the industry, the academia, and government to work on these projects. So if you're interested in this initiative, I encourage you to contact your provincial genome center to get more information. So before I pass it on to Nadia, I would like to talk about what we do at NRC. So we have a program called Environmental Advances in Mining. It's a multidisciplinary approach to tackle environmental challenges in the mining sector. We have different areas of research. We have ARDML, effluent treatment, environmentally friendly processing technologies, selenium, sensing and monitoring. We are working with mining companies and supply chain to de-risk and lower the cost of adopting environmentally friendly practices and to lower the cost associated to liability. NRC has extensive expertise in genomics and microbiology that can be applied to the mining sector. We have experts in microbiology, microbial ecology, molecular biology, hydrogeology, biochemistry, and bioinformatics. We provide services to mining companies and consultants, and we collaborate with academia and other government entities. We have some projects on biooxidation and heap leaching, biocyanidation, biodegradation of giant tires, microbial fuel cell-based sensors for water contaminants, characterization of microbiomes in tailings, and characterization of bioreactor metagenomes involved in gold recovery. Thank you. I will now stop sharing my screen and pass it on to Nadia.
All right, thank you, Julie. Can everybody hear me?
Yep.
Yes, good. Okay, so I'm going to be carrying on with the same discussion that Julie started in looking at the application of genomics specifically in mining applications. And I'll expand on a bit of background that Julie gave and then really talk about, spend more time talking about a project that Julie and I at the NRC and here at Laurentian University, where I'm located, have been working on in trying to advance the application of genomics tools in different aspects of mining challenges, whether it's waste valorization or looking at reducing liabilities. And we'll talk about how that can help provide a variety of tools for the mineral resource sector and looking at ways of improving sustainability in mine waste management. So to give an overview, and Julie presented that genomics can be used in a lot of aspects of the mining cycle, and this is also true in developing tools that are specific to different aspects. So my research program is a good example of how there is no one solution or one type of study or one type of microbiome that we need to be targeting. On one aspect, if we're looking at the valorization of mine waste, you can see in this blue column, we're looking at a number of different projects in extracting value from different wastes using different established technologies, whether that's something like heap bioleaching, passive treatment to reduce the liabilities and perhaps extract some value in some metals, but also look at less common applications and looking at potential for reductive dissolution and developing ways of processing waste that don't necessarily generate some of the downstream products that may prohibit some of the applications due to costs in treatment of different waste streams. On the other hand, and we're not going to be talking about this today, but looking at different groups of microorganisms that can be used to apply remediation technologies to either dry wastes or liquid waste, looking at the microbes that can help establish vegetation on tailings, for example, or the microbes that can be used to accelerate wetland treatment and the stabilization of metals in wetland soils. Now, the third column is an important one, and one that Julie highlighted as well, in that scientists like myself and Julie are having a lot of discussions with the mining sector, and it's good that we now hear that words like genomics and microbiology and biotechnology are not as foreign a concept, and it allows us to look at the application. What are the next steps in not only understanding how these tools work, but how they could be effectively applied in different mining scenarios. So that is something that I actually include in my research program, in talking about these technologies and how we move them forward. So looking at where we're situated, this image is from the Coppercliff tailings area in Sudbury, Ontario, but it could be many other mine sites. In looking at the liabilities presented from the different waste streams that are produced in mining. And those we know quite well as liquid and solid waste streams, acid mine drainage, tailings, waste rock, and slag. And on the component of understanding the processes, the geochemical processes that drive negative outcomes like AMD, we really have to understand the microbes and the microbial communities that exist in them to be able to either control the processes or accelerate them if we want them to work for us. And the outcome, of course, if you're in Sudbury, is to produce more nickel and extract some of the metals that have been left over in the legacy materials, but also look at improving the effluence and the quality of materials that are being left in the environment after closure. So where does biotechnology fit in? Julie already gave some background in how we can apply it, but there's opportunities to look at the application of biotechnologies at active mine sites, but also at abandoned sites. And that is something here just as a snapshot that everybody in Ontario is aware of. We have a lot of sites. This is a snapshot from around Sudbury looking at northeastern Ontario. And each of these dots is an abandoned site. Many of them may not have tailings or other materials. They could have different physical hazards, but it just gives an illustration of the number of sites that could potentially be targeted with different tools that are perhaps less expensive to, again, either look at waste valorization or look at bioremediation opportunities to help alleviate some of the legacy issues at these sites. So what I'd like to talk about in building on what Julie had said in why we need to apply genomics tools when we're studying these systems is that we really need to harness and accelerate our understanding of the diversity of biology, the diversity of organisms that could live in these sites. And this is something that we've built a lot of the literature on in the last 10 and 15 years since the advent of genomics tools. But of course, we've been studying these communities now for several decades using traditional microbial techniques such as culturing in the lab. But genomics has really allowed us to expand our understanding by looking at the diversity of organisms and how they're adapted to different conditions. So as we know, no two tailing sites are the same. And so we shouldn't expect that the organisms living there and the communities living there are the same. In fact, they're very different and express a variety of different metabolisms and abilities to withstand certain extremes. And those could be chemical or physical extremes. And when we take that one step further, the goal, of course, is to not only understand and describe who's there, drowning people in bioinformatic data or genomics data isn't going to get us to solutions. So it's really taking that one step further and being able to harness or isolate specific groups of organisms or promote certain groups of organisms that are going to carry out the desired outcome. And in the process, we are also looking to discover novel metabolic functions or networks of organisms that might be particularly suited at targeting something like a very refractory material for extraction of gold. And that's a sort of a lead up to a project that I'll be talking about in a few slides. So as Julie mentioned, when we look at the gaps in understanding the biogeochemical processes in something like mine waste oxidation, sulfide mine waste oxidation, it really is the genomics tools that allow us to monitor these systems and see how dynamic those systems are over time. In the interactions between microbes and mineral surfaces, the interaction of microbial, or the rate of microbial activity in the production of effluents, looking at how that changes over temporal scales is critical, and it's one more piece of data that could be used in monitoring of how these systems change over time and could help highlight some key areas that might be targeted for particular management strategies. From the bioleaching side of things, and this is where I'm going to shift my talk, less so on applications in remediation and monitoring, but looking at how we can use genomics tools and apply genomics tools to advance the application of bioleaching to the reprocessing of mine waste. And that could be oxidized tailings that have been sitting in the environment for some time, or it could also be looked at as an application, an additional technology during mine waste active operating mines and generating a freshly deposited tailings and reprocessing those before they're put out into the environment and generate acidity. So the group of organisms that we'll be focusing on are the organisms that cycle iron and sulfur, and particularly those that can oxidize iron and sulfur and break down primary sulfide minerals. And in the process, by breaking down those sulfide minerals, can release metals into solution. Now, even though we're going to be looking at how genomics can be used here to improve waste valorization, the process, of course, is releasing the sulfides from the material, and the materials that remain after bioleaching are going to have less potential at generating AMD in the long term. So the caveat that I want to put on this, and most microbiologists will understand this, is that genomics tools, as good as they are still dealing with an extremely complex environment. Even though traditionally we have thought of as mining environments as sterile and low diversity, with genomics tools we have uncovered that really this is in fact a thriving system that has different microbial communities, even in something like this 30 centimeter gradient that you're looking at in a tailings deposit. At the surface, you will have microorganisms that drive and accelerate oxidation in the oxic zone. And then as you move down into the reduced zone, you have completely different groups of microorganisms that may perhaps promote reduction or could even drive reductive dissolution and still generate and contribute metal-laden effluents downstream. So when we're looking at sampling and understanding these microbial communities, we have to overlay the fact that there is a lot of inherent complexity in these systems, and we can't hang anything on any one or two or three individual species. It's understanding how these communities interact in these systems. So Julie already helped introduce the idea that these genomics tools allow us to tease apart this complexity and look at function in the environment. And yes, it does generate a lot of data, and our goal as microbiologists is to be able to tease out the important information and look at that information within the context of the environmental condition and use it to inform either the process or optimize a process. So within the bioleaching framework, I can distill out what's on that previous slide in highlighting that genomics is only one piece of the puzzle, and genomics alone without the understanding of the environment or the geochemistry is not going to allow us to take a condition or present features of a site, apply genomics tools, and hope that we're going to come out with a specific solution and then apply it to all sites. We really have to take the tools, the genomics tools or meta-omics tools, and apply that to every particular site and be able to understand the specific processes, because those are going to be unique then to the geochemistry, the interaction of the biology and the abiotic component, to then come out with an understanding of how these microbes and these communities can work in this particular mineral system. So I'm going to give an example of a project we've been working on. I selected this project because it's one that Julie and I have been working on for the last year and a half. So we have two academic partners, Laurentian University and Cambrian College. We have our industry partner, a company called BACTEC Environmental, NRC at BRI in Montreal, and a technology company, Metagenome Bio, based in Ontario. So we're going to look at how genomics is being applied to develop a process looking at a refractory gold tailings material. And you see in front of you a number of different little samples of material that we're hoping to extract gold from. And so I'm going to tell you a little bit more about these materials, how we've applied or developed a test system, and how we're using genomics to help tease apart what's happening in a bioleaching system, and how we can use the genomics tools to help identify who are the key organisms that we want to use in what could be a scaled up process. So the picture that I showed you on the left, you've seen that same site. This is a site from Sudbury, Ontario. It is an arsenopyrite deposit. We've been studying that site for a number of years and have various cultures of microorganisms that we've isolated from there, and they're very resistant to arsenic. We then have another site, a picture on the right, which is a arsenopyrite deposit. These are tailings from several decades of processing in Ecuador. And this site, as you can tell, where the tailings are being deposited up on the mountain, is at capacity. And the government in Ecuador is looking to limit this type of deposit in the environment. And so they've been looking at opportunities to reprocess this material. Now in Ecuador, there are a lot of artisanal miners that look at sites like this as an opportunity. And they're looking to extract any value that's left in these materials, but they may or may not have access to advanced technologies. And so you will see in the second picture, the fellow standing on the left is Ross Orr, the CEO of BACTEC, next to individuals that are working in Ecuador, and they're standing in front of large bags of this tailings material from that Ponce Enriquez site. And they're using antiquated processes. You can see a lot of this is being done manually, using cyanide and mercury to get at the remaining gold, most of which is refractory in these materials. So they're refloating these tailings. They're trying to get at some of this gold. Of course, as you can tell in the process, we might be exposing individuals to materials like cyanide and mercury, and the materials are not leaching very well in that process, and that material then gets redeposited into the environment. So it's an opportunity. You can see that there's quite a lot of gold in those materials still, 2 to 6 grams per ton, but the level of arsenic is also very high. So we have a human health and an environmental challenge, and something that could benefit from a different technology like bioleaching. So we were engaged by BACTEC to find better ways of extracting the gold from these particular materials through bioleaching, and work with the artisanal miners to find a better route and avoid cyanide and mercury to get at that gold. So these are the samples. This is the diversity of material that those artisanal miners are acquiring. So here I'm going to just show 7 different samples. They range in the amount of arsenopyrite, chalcopyrite present, and also quite a lot, a range of arsenic. Here I've only shown that it's up to 15% by weight, but that varies from 2% to 15%. And some of these, the really oxidized materials, the orangey yellow ones that you see are the primary materials, and then the gray ones have been concentrated. So those have been refloated, and we're looking at processing those. And that's why the gold grades you see 30 to 80 grams per ton. So we wanted to target this with some of our understanding and the cultures that we had in the lab that we knew could be resistant to higher concentrations of arsenic. And you can see some flasks in the lab. We're also using different cultures and consortia, groups of organisms from different sites. So I'm going to show you how we've set up a test system, and then you'll see how we've been applying genomics to help understand this system as we try and get at that refractory gold. So we've got a set of bench scale reactors. These are mimicking some of the conditions that would be used in a full scale reactor, but of course a batch reactor is not a perfect mimic of what would be used at a full scale, but it allows us to test a few different parameters compared to the shake flasks that I showed on the previous slide. So you don't have to focus on any of the details other than that we can set up a battery of tests on different reactors that use different blends of material, knowing that arsenic is going to be a challenge for microbes. There's toxicity that the microbial cell has to deal with. And so you have potential to blend the materials that the artisanal miners are acquiring and look at the application of different consortia. The names of the consortia don't matter, but I do want to contrast that we isolate and we think we have these consortia of organisms that should work very well in these conditions that we think they're well adapted to. But of course, the materials always come with a native consortium. These are bacteria that have invaded these materials naturally, and they come with the materials when we work on them. So we want to use genomics tools to understand how the defined consortia stack up against the native communities. And then how do these communities shift while they're in the reactor and we're trying to promote the oxidation of the arsenopyrite to then make the refractory gold amenable to recovery. So we run these reactors, we add basal salts medium so that the bacteria have a bit more food and are able to grow more rapidly. We apply routine analyses that allow us to monitor the geochemical conditions over time. And then of course we want to use these genomics tools that we've been talking about to help us understand how this process is carrying on through the course of the oxidation trials. In the end, of course, you can imagine that in the situation, in the pictures that you saw from Ecuador, the goal is not only to help artisanal miners extract the gold, but also help alleviate some of the environmental challenges and burdens that they're carrying. So we also want to be able to stabilize the arsenic in this particular application. So in the test termination, we're not only looking at gold recovery, but also looking at how much of the arsenic has been released from the primary mineral so that we can then precipitate it in a more stable form as ferric arsenate. So I'm not going to get into a lot of the specifics of the data that I'm going to show you, but I'm going to give you a sense of the variability that we encounter when we work on a variety of materials that could be blended in a reactor at any given time. So you're looking at an oxidation reduction potential graph over a number of days that we've been performing oxidation in these different reactors. And you see that the different consortia, which are the different colors that we've applied to one type of material, here we're looking at just at one blend, actually perform very differently. And so we have different redox potentials, which are going to then impact how quickly or how efficiently the primary arsenopyrite is being broken down. When we then compare two outcomes, on this slide I'm going to show you the arsenic, and then I'm going to show you what happens with the gold. For this one particular blend of material that we were testing, we had our different consortia, and on the left-hand side, you see a concentration of arsenic. We take the material after we've processed it in the reactors, and we send the residuals for toxicity testing, the TCLP testing that's carried out, to see how much arsenic could still be released from the residuals. And you will see on the left-hand side that our three consortia, Bactec, Snow Lake, Telemayu, and then ECT, which is the native consortia, all oxidize and release the arsenic into solution so that there's very little left in the residual material. Now, this is very good. This compares to the large bar that you see over 15 milligrams per liter, that arsenic has not been released. There is still quite a lot left in the solid residuals, and if that was to be left out in the environment, would potentially be leaching that out into the receiving waters. So we can at least demonstrate that through biooxidation, we are then releasing the arsenic that can then be stabilized. And when we take that one step further, on the right-hand side, now look at the concentrations in fact being much lower, we can look at the variation even at a minute scale that the blend or different types of materials are going to result in more or less residual arsenic. So on the right-hand side, all of these are below the US EPA limit of 2.5 milligrams per liter. But we also still want to optimize that even further to see what blend characteristics are going to be most amenable to the consortia or the bacterial communities that we're using, so that we can stabilize as much arsenic as possible. Now, the bigger question for companies like BACTEC and for the artisanal miners is, well, how much gold are we able to get out through this process? And these numbers may not look fantastic, and we're still working to improve those, but when you're looking at the recoveries starting at 20 to 30% using traditional methods, the bioleaching process using different consortia is leading to recoveries between 70 and 80%. We want to get those above 90%. Now, the reason we see this incomplete oxidation of the arsenopyrite that's harboring the refractory gold is that we're still trying to figure out what is that threshold for too much arsenic for the microbial community to effectively break down those minerals. And so because we have this variation in gold recovery and some variation in arsenic release, we really want to be able to understand the system and why or why not, why our consortia are operating the way they are, and why might we not getting the results that we want. And I just realized that my graph is far fuzzier than it should be on here, but this is a bar graph like the one that Julie showed. And this is how we're really using the genomics tools to monitor the reactors like we're monitoring pH or ORP over time, to understand how those dynamics change and then correlate those to the outcomes that we're seeing. So what you'll see on the very right-hand side are sort of the key iron oxidizing organisms that we thought would be really good at operating in these systems. We've added things like sulfobacillus or leptospirillum or acetithiobacillus, and you can see those colors in that 2-week bar. They are the dominant organism. You've got the blue, the dark red, the yellow. Now you will note that in weeks 4, 6, and onwards, that those primary organisms that we've added in are no longer the dominant organisms. In some cases, like sulfobacillus, you do see them carrying through over time, but in the other cases, in the other reactors, they're being outcompeted. And we can monitor now and see how the native consortia, the bacteria that come with the material, are in fact better adapted to the conditions present because of the high arsenic concentrations or the acidic conditions, and they end up thriving and outcompeting the organisms that we know actually do a good job at breaking down the arsenopyrite. So without having this information, it's very hard for us to understand what's happening in the system, why are particular organisms appearing or disappearing, and then figuring out at what conditions we see a shift from our organisms of interest to organisms that perhaps are better suited, but may not be, in fact, doing what we need them to do in breaking down the mineral of interest. So when we take the genomic information, and again, I don't want you to pay attention to the specifics here, but we look at the specific metabolic genes that are expressed or present in any of these particular reactors, and we look at pathways that might be turned on or turned off. And by using these genomics tools, we're able to see where systems are demonstrating the key pathways, like the SOX pathways that you see here, that are showing us that sulfur oxidation is happening, and it is happening because certain organisms are present and active. We can also take that genomic information and we can look at if there are issues in other nutrient cycles and carbon cycling, or we can look at the presence of genes that might be helping organisms adapt to the conditions. For example, the pie chart that you see on the right-hand side, looking at arsenic resistance. We know that this is an arsenic-dominated system, and we want to be sure that the organisms we are adding in and that are surviving are able to resist the pretty harsh conditions that are present in those reactors. So having this genomic information doesn't just allow us to see what's there and what the capacity might be, but as Julie mentioned, we want to get to the ability to monitor these systems in real time. So if something drops off, we can figure out why conditions have crashed or why the organisms that are catalyzing the reaction are not as active. So here we are, this is where we are in the process. We've gone from bench scale tests to get organisms. We've now applied them in reactors, but we really need to do several rounds and iterations of this test work, use the genomics to better optimize the conditions for the microbial community that we know will perform best, to push gold recovery higher, to increase arsenic stabilization. And then the next the next steps will be to scale this up more towards what would be carried out in the field, so in Ecuador or elsewhere, and ultimately use that information to build a demonstration facility. So where we are right now, sort of between steps 2 and 3, the genomic information is still very large and very heavy, but it's allowing us to streamline the process so that we can get down to a handful of organisms and conditions that we can monitor so that a full-scale plant can use these tools to monitor just like they do other conditions. So Julie already introduced one of these newest technologies, the MinION sequencer, and our next step will be to introduce this type of technology to allow us to monitor these systems in real time. It really doesn't help to have data 2 weeks after the fact if we're running a process, and the inclusion of genomics as a regular tool for mining operations will only come if we're able to get that data as quickly and as efficiently as other parameters. So our next step in working with Julie on this, looking at how we can take the understanding of the reactors we've already run, the communities that we know are present and may or may not be struggling over time, how can we get that data quickly to then change the conditions in the reactor, perhaps add in organisms or particular additions to help particular organisms survive, and allow the reactor to then continue at an optimal level rather than allow it to crash and then bring it back up. So this is our challenge right now. So we're actively working to make this easier so that when projects like this are complete, we can demonstrate that genomics can be streamlined in these types of applications and hopefully de-risk some of the concern around how challenging this technology might still seem to non-microbiologists. So just to summarize, I think Julie already highlighted that the genomics applications and genomics tools are rapidly advancing, and individuals like myself and those at the NRC and our other partners are really trying to demystify molecular technologies and streamline how we're using those tools to feed in our understanding of different applications, whether it's for AMD prevention, valorization of wastes, or downstream processes in treating water and understanding wetland microorganisms, for example. I think we all look at the opportunity to use genomics tools in bioleaching and bioremediation as an addition to an existing tool set. We're not saying it's going to solve all the problems, but I think, as Julie said, it would be important to have an additional voice of microbiologists on mineral extraction or environmental teams looking at ways of reducing some of these liabilities, improve metal recovery, increase waste reduction, and look at opportunities of remediating sites like abandoned sites. So one of the projects we're working on, and this has come about as a request to scale up a lot of technologies. This is a capacity that is increasing, I think, across Canada at a number of sites, both academic and private and government initiatives. But of course, we have to move from bench scale to demonstration scale. So we're currently working on developing a Center for Mine Waste Biotechnology here in Sudbury, looking at being able to scale up things like I just showed you with the reactors to a larger scale in biomining, but then also creating facilities where we can use and demonstrate how genomics and biotechnologies can be applied in soil and phytoremediation, in treatment wetlands, and also looking at full-scale demonstration bioreactors. So that is an overview of some of the ways that we're trying to apply and advance and de-risk genomics in mining applications. And I think collectively, I see this more at conferences and discussions, that it's less of an afterthought, and we're hearing more about biotechnology applications as part of the mining cycle, not just as an afterthought after closure. And so it's a very encouraging trend that I think we see in Canada moving in this direction of broadening the tool set that we have in dealing with legacy and ongoing issues in mining. So with that, I'd like to thank you and show some happy faces of the people that carry out all this research in my lab, and our partners, and of course all of our partners at the NRC, at other government institutions, academic institutions, and private sector as well. So with that, thank you.
Excellent. Thank you very much to both Nadia and Julie. It was a really interesting webinar, and I appreciated both of your times. Does anybody have any questions or comments for either Julie or Nadia?
Can I just, it's Karen, can I just suggest that you unshare your screen so that we can see everybody and invite everybody to come on camera?
Yes, I will.
Then we can see people waving if they have a question.
Find the button. No, stop share. There we go.
There we go.
Good.
If people are not too shy, they can come on camera. You can tell we have a bunch of engineers online. We won't bite.
Well, we are actually hoping that, you know, all of the experts that have been traditionally involved with the mining operations, that they will open up to new tools, to new technologies, and to also discussions with new experts like microbiologists and microbial ecologists. So hopefully now we have mining engineering students on the webinar and other people that we're trying to reach out to. So hopefully things will change for the best, and everybody will be able to collaborate, and everybody will benefit from these collaborations, basically.
Janice, did you have a comment or a question? So you raised your hand on Zoom.
Can you hear us okay?
Yep.
Okay, we're a group of us here at Camet Mining listening, so great presentation. And Dr. Sean Langley here in my group has a question.
Hi, either to Julia or to Nadia. Can you provide a little bit more information specifically on how the MinION works for us?
Julie, you want to take that one?
Sure. So basically, well, you have to extract the DNA from the sample, but there are some very simplified DNA extraction protocols, very, very short, very rapid. And then you need to, we call that preparing the library. So this means that the DNA molecules, they have to have some special adapters linked to the molecules so that they will actually be compatible with the sequencing chemistry. So these are the same steps for all of the different DNA sequencing technologies, except that for the MinION, they developed some very easy and fast to use kits. So it can all be done under 2 hours. Extracting the DNA, preparing the library, and then you load your sample. Nadia's slide was showing like the pipette, you know, pipetting the sample on the DNA sequencer. And then you plug the MinION on your laptop, and you have the DNA sequencing data that's showing up in real time. And when it's completed, then you can, if you were analyzing multiple samples, you can demultiplex the data set. So each sample during the library prep process was given a particular index to be able to sort out the reads that are associated to each of the different samples. And then you have the analysis step. So this is the step that needs a bit more work to be able to really use the MinION in real time. This is something that Nadia and I would like to work on with our team of bioinformaticians at NRC. So we need to find a way to make this data set analyzed very efficiently so that we get the information as close to real time as possible. So for now, the lab steps are pretty much optimized. It's pretty quick to prepare the sample and get it analyzed. So it's really the analysis that we really need to focus on for now.
Okay, great, thanks.
We have one more question here, if that's okay.
Sure.
I have one question about this MinION. Can it be used for also metagenomics, or it's just for metabolic coding? I mean, the entire genome is being sequenced real time, or just a particular part of the genome?
Both. You can use it for a specific gene like the 16S if you're really only looking at characterizing the microbial population. So you can do amplicon sequencing, 16S, or any other gene that you would be interested in, or you can do full genome. You can even do RNA sequencing. It's very flexible, but for sure, analyzing data from metagenomes versus amplicons, it's gonna take a lot more resources and time. So like I mentioned, this is something that we really want to work on. So we want to be able to have real-time results for metagenomes as well as for amplicons.
Yes, Asma, that was a great question. And I think the RNA-seq part, so being able to look at the expression of particular metabolic pathways at a given time, it's still challenging. There's a few examples where direct RNA-seqs worked well with the MinION, but again, it's going to take more projects, more applications, but Oxford Nanopore is working hard to advance that.
Also, just to mention, the MinION is generating a bit less data than, let's say, the other platforms by Illumina, like the MySeq or the HiSeq. So it's not replacing these technologies, it's complementing these technologies. It's filling a gap that the other technologies had left. And it's not meant for every project. It really, whenever there's a project, you need to really sit down and look at all the different DNA sequencing options to make sure that you choose the right one for your project. And one of the advantages of the MinION, aside from the portability and the ease of use, is that it creates much longer reads. So the genome assembly in the end is a little bit faster, a little bit easier.
Thank you.
I have a question, and I hope I didn't miss this because I was sort of hopping in and out, but is there special needs or requirements or ways that you need to work with taking the samples to make sure that you're getting the right conditions for all the further testing in the lab that are different than, say, other sampling protocols?
Julie, I'll go first on that one. You can add all the methodological challenges we have. So, of course, your sequencing is only going to be as good as the DNA that you get out of it. And hopefully the DNA that you're getting is what you're trying to look at, not all the other DNA in the environment around your sample. So absolutely the protocols on handling samples, on extracting DNA, on making sure that you have controls, negative controls, to be able to remove any background contamination before you make any conclusions about the data that you're getting out, are going to have to become sort of standard operating procedures in handling and sequencing. Now, of course, we're still working through a lot of those. Genomics as a whole in different disciplines and health and environment being able to tell the difference between what might have just been on your swab or not extracted from your sample is something that has to be carefully managed so that you're not interpreting bad data.
And just to add, we also work with replicates. Typically, we work with triplicates to make sure that the result that we're getting is a real result. It's not like an outlier. So when you have triplicates and they all show the same microbial profile, for instance, or the same gene functions, then it's a bit more robust.
Thanks. I see there's a question or two in the chat that maybe we can say out loud for everybody.
Yes, I answered one in the chat. So from Abu was asking if bioleaching works well in arid environments. And in fact, a lot of the current examples that are operating are in arid climates, warmer climates. Examples that I provided, like in high altitude mines in Chile. There's examples of some in the Middle East, as well as in Western Australia. And the challenges, of course, at applying these types of applications in a place like Canada is we have a very dynamic climate, and the switch from going to very cold or very warm, dry and wet is challenging for bioleaching operations that are open. So the heap leaching operations, much less of a challenge if we're taking the bioreactor route where we can control the conditions. But you've asked if there's any references. Of course, there's actually quite a big body of work from the existing operations, and that's helping inform what we might do in colder climates. And then there is one example of bioleaching in heap leaching applications from a cold climate. So that's at the Terrafame site in Finland. That is actually giving us some interesting insights of how these particular iron sulfur oxidizing organisms work in a colder climate. So it's an exciting time to be working in this field, because you see just how being able to monitor the microbial community can inform what happens in these systems in different climates.
Does anybody else have any questions or comments? Okay, if not, if everyone is good, then I think we'll end the webinar. And thank you again very much to Julie and Nadia for taking time out of your days to share this with us. Just a reminder.
Thank you for listening.
Thank you.
And look forward to hearing more, perhaps more of this content at the CIM conference. If anybody has any submissions they want to make, deadline's next week, so get them in there.
Will.
Okay. Thank you everyone for attending.
Thank
you. Bye.
Bye.